I've got some slides. Oh, that was loud. I've got some slides to take us through today, focusing on all the core material. Happy to take questions at the end. Equally, if I'll try and keep my eye on the chat. So feel free to send some messages there as well. Okay, so first of all, here is your syllabus. Hopefully, that's not a surprise to anyone. We'll try and run through everything on the left. I've tried to kind of do a traffic light system. So in the top right of the slides, I've tried to put a green circle if it's core material and then a red circle where it's not core. I've really tried to keep this as core focused as possible. I've just stuck a couple of things in about some of the key experiments and some clinical models, which might be useful to keep in mind while you're thinking about this. Okay, so let's get started with the resting membrane potential. So this is a very basic model of a cell. You should appreciate that there's this unequal distribution of ions across the membrane. So crucially, you've got sodium chloride and calcium ions with a higher concentration in the extracellular fluid than in the intracellular space. And you have a higher concentration of potassium ions and negatively charged anions inside the cell. These anions are pretty much amino acids and proteins which are being produced by the cell and they're staying in there. Oops, changing slide. There we go. So some key membrane proteins are involved in setting up the resting membrane potential. The first one is this sodium-potassium pump, or the sodium-potassium ATPase. And that's responsible for an active transport of two potassium ions out. It's two potassium ions in for every three sodium ions out. This helps to establish a concentration gradient for the sodium and the potassium. This is actually quite a minor contribution, though, to the overall resting membrane potential. Most of it is done by these constitutively active potassium leak channels or inward rectifier channels. These allow potassium to follow its chemical gradient and diffuse out of the cell by facilitated diffusion. But it leaves the anions inside, thus generating an electrostatic gradient because we have this concentration of the negative ions inside the cell and the positive ions outside the cell. The structure of the leak channel is such that only potassium can get through, so it's very selective. Particularly, it's worth thinking about how it does. It allows potassium to get out of the cell, but not sodium. It does this. There's four carbonyl groups at the intracellular opening of the channel, and the sodium ions are a bit too small to interact with all of them. So the sodium ions can't get out, but the potassium ions can. We get an equilibrium reached where the concentration gradient for the potassium to leave the cell by facilitated diffusion is equalized with the electrostatic gradient for the potassium to go back in. And we call that the Nernst equilibrium potential for potassium. That comes to about minus 90 millivolts. That is not obviously the resting membrane potential for the whole cell. That's just the major contributor. The rest of it is done by this similar process for an equilibrium potential for all of the other ions. So a little bit more on that and how it actually works. So the Nernst equation, which is shown on this slide, is the equation to bring together all of the different ion equilibrium potentials and work out what the overall resting potential should be. So we can, this is looking at one particular ion. So we're looking at the one on the left, it's generalized for any cell. But because most of them are constants and because the temperature in a mammalian cell should be pretty constant at physiological conditions, you can actually simplify it. So there's this equation on the right. I wouldn't expect you necessarily to be able to need to reproduce this for your exam, but it is worth knowing where these numbers come from. Yeah, you won't need to derive where it comes from. I hope that's sort of a bit of an intro to where the resting membrane potential comes from. This summary slide tries to illustrate that with diagrams. So overall, we end up with a resting membrane potential of minus 70, which is caused by polarizing potassium efflux currents, mainly from this inward rectifier or potassium leak channel. It's leaving down its electrochemical gradient, and there's a small role for this sodium-potassium ATPase. The slightly more complex model, which they mention on the right side of the syllabus, is this Goldman-Hodgkin-Katz model. This is where they've tried to further develop the Nernst equation to give you a more comprehensive understanding of the resting membrane potential. So what they say here is we can work out what it should be by looking at the difference in the permeability and the concentration gradient of potassium, sodium, and chloride. Now, because the permeability for potassium is the greatest, it is the major contributing ion. In terms of clinical correlates, hypo and hyperkalemia are two very common electrical abnormalities that we see in practice. Both of them have broad-ranging effects on electrophysiology because of the influence that they have on the resting membrane potential. In hypokalemia, so that would generally be when the potassium in the extracellular space gets below 3.5 millimolars per liter, we get a hyperpolarization of the resting membrane potential. A patient might present with fatigue, leg cramps, weakness, and constipation. You can correct that with a potassium infusion. Hyperkalemia would be the opposite. So you get a depolarization of the resting membrane potential. So you get muscle weakness and paralysis, palpitations, nausea, vomiting. And we correct this with calcium gluconate and then an insulin glucose infusion to try and move some of that potassium back into the intracellular space. It's worth thinking broadly about how you might get these. So in clinical practice, there are lots of different ways that this might actually happen. So hypokalemia, for example, you might get from overdiuresis of a patient or from nutritional deficiencies or, I guess, vomiting as well. Whereas hyperkalemia would be more likely to come from something like a kidney injury where the patient is unable to excrete potassium. Okay, that's everything. I can't really actually see the chat, so I don't know if anybody's asked any questions yet, but feel free to shout out if there's any questions about that. Otherwise, we'll move straight on to action potentials. Okay, great. So an action potential, we're going to be thinking about the ionic mechanism for an action potential, which was sort of all characterized by Hodgkin and Huxley. An action potential, I'm sure you're all aware, but it's the rapid change of the voltage across the membrane. They're described as all or nothing. So a nerve or any excitable cell will fire when they reach a threshold potential. When that threshold is reached, it causes voltage-gated sodium channels to open. These are responsible for the upstroke that we see here on the left side of the diagram. When the voltage-gated sodium channels open, the sodium can get into the cell, so it starts to depolarize. And when it reaches a certain threshold, then the voltage-gated potassium channels start to open. Over a time course, the voltage-gated sodium channels inactivate. So we'll have a little look in a minute about how that actually works. But the voltage-gated potassium channels remain open, and they're responsible for repolarizing the cell. So we see the membrane potential come back down. And then we get an overshoot. So it goes a little bit further than you would expect beyond the baseline. And then the voltage-gated potassium channels close. And eventually it returns back to its resting potential. There are two refractory periods which you need to be aware of. So the absolute refractory period is the time period when no action potential could fire again because all of the sodium channels are inactivated. Whereas the relative refractory period is this period where the sodium channels could be activated again, but we need quite a lot of impulse to get it up to the threshold potential because the rectifying potassium channels are still open. Propagation is ensured by the fact that we have these local currents. So in a excitable cell, the action potential will only travel in one direction because as the action potential moves past that particular local current that it's just been in, the voltage-gated sodium channels, which are closed, remain closed for that absolute refractory period, and then it takes quite a lot to fire another action potential. So in that way, it's always going to keep moving sort of away from where it was, if that makes sense, because it would, because we're not going to get back to that threshold potential again. So channel states, this is not on the core syllabus, but how do the voltage-gated channels actually work? So they're closed constitutively at the resting membrane potential. Depolarization will cause the voltage-gated sodium channels to open, and that literally means a gate will swing open. So there's some electrical interaction as the membrane depolarizes, which changes the shape of the protein and the gate opens up. With time, another gate closes, so it's two different ends of the protein. It covers up the, I guess, the efflux area or the extracellular part of the protein, and so the sodium channels can't get through the sodium can't get through the channel anymore. Differences in the kinetics of channels will obviously affect how long it takes for this to happen, but that will depend on a channel by channel basis. Okay, so we studied this by looking at by using clamping experiments. We'll talk about the classical one in just a minute with Hodgkin and Huxley. It's worth having a think about what these different clamping techniques are, though. So there's voltage and patch clamping. We've tried to summarize them concisely here. I think one way of thinking about it, though, would be that the voltage clamp technique is really good for measuring the function of like one individual ion channel, whereas the patch clamp, sorry, other way around. The voltage clamping is good for looking at an area, and then the patch clamping, we're looking at single channels because you suck it up into the patch clamp apparatus, and you can measure just the ionic flux across that particular part. So the classical experiment which demonstrated the ionic mechanism is the Hodgkin and Huxley series of experiments, which got them the Nobel Prize. They used the giant squid axon because it is very large, 0.5 millimeters in diameter, so they were able to directly insert micropipettes into the axon and then perform voltage clamping to see how the voltage changed when they when they changed the concentration outside of the neuron, and then when they added different toxins. So TTX, TEA, and ion replacement, like I said. We'll talk a little bit about what those different toxins are when we get into thinking about the NMJ, but it's worth thinking that they're quite similar in their effect to a local anesthetic. Okay, so conduction velocities. This is back to our core material. Conduction velocity changes a lot depending on what type of nerve fiber you're looking at. You might consider thinking about why that is for an essay. So we have different types of fibers which are responsible for the transmission of different types of information. So you have your alpha motor fibers, which are pretty fast. The fastest ones are your proprioceptive ones. And then you have your unmyelinated type 3 and unmyelinated C fibers, which are slower, and those are really responsible for nociceptive transmission. So pain. Saltatory conduction is a major contributor to how fast action potentials will be propagated through a particular nerve fiber. So saltatory conduction is this idea that we have areas of a axon which have a Schwann cell wrapped around it. This increases the resistance in those areas which have the Schwann cell, and there's relatively lower membrane resistance in the nodes of Ranvier in between them. This is useful because it means that you don't have to have those local currents all the way along the axon, but rather you can have them just in the nodes of Ranvier and then diffusion across the high membrane resistance areas, and then just another local current at the next node. In terms of ion channel distribution, you get a very high density of expression of the sodium channels in the nodes and in the area just next to the myelin sheath. You get a lot of the potassium channels. So the way that it's actually set up is they will spatially separate out the sodium and potassium channels to try and optimize how quickly this can happen as well. The myelinated ones are always much faster than the unmyelinated nerve cells. Next is the length constant. So this is a measure of how far along an excitable cell a signal is able to travel before decay. This is determined by this membrane resistance and the internal resistance. So like we were just saying about the myelination, we've got areas where there's going to be more membrane resistance, so essentially less ability of ions to cross the channel, cross the membrane, which will be largely determined by ion channel expression in those areas. And then lastly is the time constant. So this has been defined here as the time taken for a nerve to reach 63% of its final change in potential. And that's fine. But generally, I like to think of that as how quickly does an excitable cell respond to changes in its membrane potential. That's a product of the resistance of the membrane and the capacitance of the membrane. The smaller your time constant, i.e. the faster it can respond to a change in the membrane potential, the faster it will be able to conduct a signal. Okay, and hopefully this is summarized here. The other thing to think about is that I didn't mention was the diameter of the fiber. It was back a couple of slides on that table. But obviously, if you have a thicker fiber, then it's going to be able to have faster diffusion going through it, as you would expect. The temperature will also have effects on the rate of the diffusion. So having higher temperatures will, in theory, increase the rate of transmission, but obviously that only goes up to a limit. Okay, so demyelinating diseases are a good clinical correlate here. So multiple sclerosis is an autoimmune condition where the myelin proteins are attacked by an inflammatory process. You have very variable constellations of symptoms in multiple sclerosis, including numbness, weakness, tingling, fatigue, depression, double vision. Quite often, multiple sclerosis will initially present with some kind of optic neuritis picture. So you might get an episode where someone describes going blind or getting double vision, and then you can have flare-ups and times when it goes a bit quieter in between. It's also quite hard to track where multiple sclerosis flares will be, so sometimes the presentation of the disease can move around a little bit. Guillain-Barré syndrome is where T cells and autoantibodies produced against peripheral proteins cause axonal damage. So you end up with a flaccid paralysis. This can be very serious and require ITU intervention for ventilation if you get paralysis of the diaphragm. And a little bit there about how it might be treated. Okay, that's everything I had to say about action potentials. Does anybody have any questions at this stage, or shall we move straight on to the NMJ? Okay, great. So the neuromuscular junction is how this signal gets transmitted from the nerve fiber across the synaptic gap into the muscle cell. So we've got our action potential. It's gone all the way through the nerve axon now, and it's arrived at the end of the cell, the presynaptic bulb. When it gets there, we have another voltage-gated channel. In this case, it's the calcium channels, which open as they're depolarized. Because of the concentration gradient of the calcium, it will enter the cell and it binds to a snare protein, or otherwise known as synaptotagmin 3, I think. So the snare protein causes a fusion of vesicles, which are already produced and ready in the end of the nerve fiber to fuse with the membrane. So the snare protein, when the calcium binds to it, literally just moves the vesicle to fuse with the cell membrane. And then when it fuses, acetylcholine is released into the synapse or the neuromuscular junction, and it moves across by diffusion. Acetylcholine then binds to nicotinic acetylcholine receptors, which are expressed in the muscle endoplate. You get two molecules of acetylcholine for every receptor, and on binding, it causes the receptor to open into an ion channel, and it allows cation influx almost exclusively the sodium just because of the concentration of the extracellular fluid. Acetylcholine, it's important for your exam that you understand sort of the life cycle and how it's produced, stored, and released. So choline acetyltransferase catalyzes this reaction between choline and acetyl-CoA. So you end up making acetylcholine and CoA again. This is then stored, like I said, in vesicles. There's an antiporter protein which is able to take up the acetylcholine driven by this active transport process. It's released when synaptotagmin 3, not 1, not 3. So the snare protein binds to the vesicle and stimulates exocytosis. And then, as I said, two of these acetylcholine molecules bind onto the receptor at the alpha subunit and cause the port to open. There's then an acetylcholinesterase, so an enzyme which is expressed at the muscle membrane, which cleaves the acetylcholine into acetate and choline so that it won't just continually keep activating the receptors. And then it can be reuptook by a presynaptic choline transporter. The receptor itself is a pentameric membrane protein. It has this central pore, which, as I said, is permeable to cations. It's not too important to understand all of this about the subunits, but just as a matter of interest, there are two alpha, one beta, one gamma, and one delta subunit in muscle, but that changes depending on where this receptor is being expressed. And the acetylcholine is binding at the interface between alpha and delta epsilon subunits. There is good crystallography evidence of this, which I haven't included in these slides, but if you were looking to do an essay on that, that might be something worth reading about. Okay, and then it's a good way of thinking through this, the physiology of the neuromuscular junction, is to look at the pharmacology. So we can look at how this transmission process can be blocked. There's lots of different drugs and toxins which target this. It's very well conserved, the NMJ setup, so a lot of natural toxins will target the NMJ. One thing I think I found hard in first year, or I probably found a little bit confusing, was the difference between non-depolarizing and depolarizing neuromuscular blockers. So the non-depolarizing ones are things like vecuronium and tubocararine. These are competitive antagonists, so they are competing for the binding site on the nicotinic acetylcholine receptors, while the depolarizing blockers are an agonist. So just highlighting that difference. Other ways that we can target this transmission is by directly inhibiting the release of the vesicles. So that would be your botulinum toxin, which breaks apart those snare proteins. You can also have acetylcholine esterase inhibitors like neostigmine, and to reverse the vecuronium or rocuronium, they're very similar. They're both non-depolarizing blockers. You can give someone something called Sugmadex. There are other toxins which aren't, all of those ones on the left are included on the core syllabus. The ones on the right aren't, but black widow venom, elatroda toxin, forms a calcium permeable pore in the presynaptic membrane, which obviously interferes with the vesicular transport as well. And W-chronotoxin inhibits the voltage-gated calcium channels, specifically the N-type ones, which are expressed in the presynaptic bulb. A clinical correlate for this is myasthenia gravis. So this is where we have an autoimmune destruction of the nicotinic receptors. Classically, this presents with ptosis, so drooping of your upper eyelid, diplopia, so double vision, dysphagia, so difficulty in swallowing, and dysarthria, which is difficulty speaking clearly, all of them caused by muscle weakness, essentially. Classically, this worsens throughout the day, which is a bit of a hallmark of your immunological conditions. It's treated with acetylcholinesterase inhibitors, so the one they've given here is pyridostigmine. And you usually give that with atropine because if you're inhibiting acetylcholinesterase, you will get a lot of muscarinic effects as well as improving transmission at the nicotinic receptors. Okay, I hope everyone found that helpful. How are we doing for time? Doing very well for time, actually. That was a lot quicker than I thought. Are there any questions before we move into doing some MCQs? Not sure how I look at the chat.
There are no questions in the chat, Finn, that I can see.
Okay, great. Thanks. Fine. So we'll move on to do some MCQs now. If you scan this, hopefully it should bring up a set of Slido questions. You can let me know if there's any problems with that. Oh, we've got one. Someone's asked me a question. So I've had a question about what are nodal NA channels. Nodal sodium channels are sodium channels which are just expressed in the nodes of Ranvier. So it's just that nodal is just a description of where they're actually being expressed. So if you have saltatory conduction, they will concentrate the expression of that protein in the nodes, obviously because that's where they'll actually be needed for the ionic conduction across the membrane. Okay, another question about the Nernst equations. So let's go back to that slide. So this is the, I'll just quickly talk through the Nernst equation in a little bit more detail. This is obviously for letting us work out what is the ion equilibrium potential for a given ion. The way we work this through, so R is the molar gas constant, which you will have learned about in A level when you're studying, what is it again? What do they call it? The ideal gas equation. Temperatures in Kelvin. So that should be pretty constant for your mammalian cells. And then there's a Faraday constant. Ion valency is going to be either +1, -1, +2, essentially, for all of the ions that you would be considering here. And the way to tell which way around you put the ions on the fraction is you see how it says xi over xo. I would be inside and O would be outside. So the intracellular one always goes on the top and the extracellular one goes on the bottom. There's one more question about it's about chlorine. So you would do the same thing, but essentially because the negatives would cancel, it would appear that it would swap, if you see what I mean. So um, purely because of the way that logs work. So if you wanted to, you could write it the other way around and ignore the negative at the start of the simplified Nernst equation for mammalian cells. But if you plugged it in normally, it should work normally. Has anyone been able to get onto the Slido? I haven't actually used it before myself, so please give me a message if you've been struggling to get on there. Slido says no active polls at the moment. Let me have a quick look at that. Sorry, guys. Yeah, it would help if I hit launch. Okay, have a go now. Hopefully it's working now. Okay, I think everyone's voted. So it is the potassium ion which is primarily responsible for the resting membrane potential. So if we think about that in terms of the Goldman-Katz longer equation that we talked about, Goldman-Hodgkin-Katz, I think that's another one. So the reason that potassium is going to be the main contributing ion is essentially down to its permeability. Okay, so because we have those constitutively active potassium leak channels, potassium is the most important ion in controlling what the overall resting membrane potential is. It's worth saying, so the resting the equilibrium potential for the potassium ion is about 92, minus 92. Um, so even though the resting membrane potential is not actually the potassium one, that's just because it's being pulled up a bit by the fact that both sodium and chloride have higher Nernst equilibrium potentials. I hope that makes sense. Basically got that right anyway. But okay, next question. How do I do that? There we go. Okay, then we've got everyone's votes there. So hypokalemia will hyperpolarize the membrane. So if we have a higher concentration of the potassium outside the cell, then we'll, what's the best way to explain this? We've got a little bit of confusion on that. So if we have a hypokalemia, because of this Nernst equilibrium potential, it's going to increase your resting potential, i.e. it's going to make it more negative because of the effect that it'll have on the concentration gradient. Okay. Okay, great. So the absolute refractory period is the period when another action potential cannot be generated. And that's because the voltage-gated sodium channels are closed for a time period. The period when an action potential can be generated with a stronger stimulus would be the relative refractory period. Okay. So the areas between the nodes of Ranvier will have much increased membrane resistance because uh, you're not going to essentially have any transport of the ions across the membrane where the myelin is. A, because we're going to have nodal expression of the channels, but also just because it's an insulator. The myelin sheath will actually decrease the capacitance of the neuron in the areas that it's covered. Um, yeah, so why? How are we explaining that? I didn't go through a definition of capacitance in this, did I? So capacitance is just how well can it store the electrical charge. It it won't necessarily increase the capacitance because the well, I suppose maybe I've worded this question poorly because in the internode spaces it will decrease the capacitance, but in the node in the periods, yeah. Okay, I worded this question poorly, so apologies for that. But it will increase the membrane resistance between the nodes of Ranvier, was what I had in mind. It will overall decrease the capacitance of the neuron because it's separating out positive and negative charges more by having an increase in the distance where the nodes are. But apologies for that. What's the role of the synaptotagmin in neurotransmitter release? Okay, so synaptotagmin is that calcium-sensitive snare protein that we talked about, which will cause fusion of the vesicle with the membrane. The protein responsible for transporting acetylcholine back into the presynaptic neuron, well, it gets broken down by the acetylcholinesterase, and then the products of that can be reuptook by a transport protein, which is a depolarizing neuromuscular blocker. Yeah, great. So suxmethonium is our depolarizing neuromuscular blocker. So it's depolarizing because it's acting as an agonist at the nicotinic receptor. So it binds onto it and opens it up and keeps it that way. The tubercularium and the vecuronium are both non-depolarizing neuromuscular blockers. They're acting as competitive antagonists for the binding site. And then the botulinum toxin is affecting vesicular transport. Which treatment would I use to manage myasthenia gravis? Yeah, perfect. See, clinical stuff's much easier. Okay, I think there's a leaderboard. Okay, perfect. So, I hope that's helpful. If that's prompted any more questions, I'll stay on here with the chat, so people feel free to send me more questions as you have been. Um, Otherwise, Anya, is there anything you wanted to add?
No, I think that's good. I think if we just leave the end slide up that has the feedback link, so if you scan that QR code, we will send over the copy of the lecture slides and you can get in contact with us if you need anything else.
Great. Thank you all for coming. I hope that was helpful.